Okay, our next topic is prokaryotic structures. And within structures, we're going to be talking about the overall structures, shapes, sizes, and then the constituent parts, basically of bacteria. So this is called prokaryotic structure, but it's really about bacteria mostly, and a little bit about archaea. And this same slide set is going to be for both today and the next lecture. It also includes motility in this slide set. This is a depiction of how we classify prokaryotes based on their overall shape. The ones that we're going to work with the most in lab are coccus, which is spherical, rods, which are rods. These can also be ovals. So a rod is anything that's slightly elongated from a sphere. And there are certain bacteria that we work with in lab. You have to watch out. Is it actually coccus or is it a short rod? And I'll point those out as we go. Spirillum, spirochete. These actually have a true helical twist to them. Spirillum are just kind of can be curved. They're not helical in a 3D sense. And sometimes these used to be called vibrios. They're now called spirillum. But if you've ever heard of vibrios or Vibrio cholerae, the causative agent of cholera, that is a spirillum shape. This group is generally being used to study development in bacteria. So when I talked about forms of life can have development where they form polarization or parts or regions. These are single cells and they have regions and they show development. And then filamentous bacteria are sort of long and elongated. We're not going to study those as much in our class, but you do need to know what coccus is and what rod is and be able to recognize it. A couple of other things I want to put on here, which are prefixes. Strepto and staphylo. Hunter, what is strepto as a prefix? What does that mean?
Chains.
That is a prefix meaning chains. What about staphylo? Cluster. So you can have clusters of cocci. So this is the singular, cocci is the plural. And if you have cocci that occur in clusters, they're known as staphylococci or staphylococcus. If you have rod-shaped cells that occur in chains, that could be an example of streptobacillus. You don't really get staphylobacillus, but you can have streptobacillus, streptococcus. And if it has that, it's in chains. One other thing that I always forget to say, but it is really critical, are the designations for what it means if you say bacteria versus bacterium. Bacteria is plural. So I always forget to say this, and then I get papers and stuff written about the bacteria acted like they blah, 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 blah. That's correct. If you talk about one cell, it's a bacterium. So please watch out for that as you go. Also, remember, while I'm on this topic, your way that you write scientific names of these. So for example, genus name capitalized, species name lowercase. And this whole thing should really be in italics if you're typing it. If you're writing lab homework, or if you're writing notes, or if I'm writing on the board, I'm not going to write it in italics, but the minute that you type it, it needs to be in italics. So please remember that. There are also certain abbreviations. If you're doing an abbreviation, uppercase, genus, period, and then lowercase, all lowercase species name. And if you're typing that, it should still be in italics. So that's the general naming and cell shape and cell arrangement. Two other terms. These are going to come up in your lab homework. The term morphology and the term arrangement. Morphology is just the cell shape, and that's what this slide is showing. What are the possible shapes of bacterial cells? That's just the shape. The arrangement is the grouping. So are they chains? Are they clusters? Pairs? Sorry. Sort of say diplos. Diplococcus. Pairs of coccus-shaped cells. Or are they single? There are some bacteria that do not stay attached once they divide, and they just appear as single cells like these. All of these actually are appearing as single cells in this picture. Generally, prokaryotic cells are smaller than eukaryotic cells, but not absolutely always. So this chart, and whenever you have a chart for class, it's usually not that I want you to memorize the whole chart. In this case, I want you to see just generally the sizes. So a normal sized bacterium might be Escherichia coli, 1 by 2 microns. 1 by 2 microns. And they're setting that as sort of a standard for cell volume. Some of the very tiniest bacteria, like Mycoplasma pneumoniae, it actually lives inside other cells. It's almost a parasite of cells. It can be way tinier than that. But there are some truly enormous bacteria that are actually, you could see it with your naked eye. It is so big. That one especially. That's the current leader. Thiomargarita namibiensis. So it was found in Africa. It has something to do with sulfur. That's the thio part. Margarita, I'm not sure. I'm not sure where that came from, but it's enormous. So some of the biggest prokaryotes are bigger than the smallest eukaryotes. So just being tiny is not all prokaryotic cells are tiny. And then you could also quiz yourself, what are their names? There's a thio, Staphylothermus marinus. You can guess things about them just knowing their names. Okay, this part, I didn't assign you to read about plasma membranes again. So I am going to just briefly review them, because some of the plasma membranes for microbes are completely different than what you've learned about before. So this would be a normal plasma membrane. You've got glycerol base structure with fatty acids coming off of it, phosphate group coming off this end, and then an additional chemical group here. So that gives you hydrophilic heads, hydrophobic tails. The tails associate together to exclude water because they're hydrophobic. Does that ring a bell? I feel like schools cover this over and over and over. But just to review, that's what we think a normal plasma membrane looks like. And here is an electron micrograph of it. These are the head groups, tails in between. You can visibly see it with electron microscopy. Also review that there are a lot of proteins in a plasma membrane. And this amount of protein is actually a realistic depiction. When you first learn about membranes, it's like they don't draw in all these proteins, but there's a lot of proteins stuck into the membrane. Every membrane has two sides, correct? It's separating something outside from something inside. Okay, here's the start of where it tends to be different. So for us, what we have learned about as a normal membrane, what you might not have, probably did not study, is that there is an ester, chemically, an ester linkage within those membrane lipids. So the R group here, this would be the beginning of fatty acid tail. And then there's the phosphate group that provides the polar head. Ester linkage. In domain bacteria and domain eukarya, they both use ester linkages in their membrane phospholipids. Archaea, part of the reason that they are designated group Archaea is because they have an ether linkage in their membrane. Which doesn't, it's like, well, it's still oxygen. It's just missing the carbonyl group there. That's not a big deal. It is a big deal. That is one of the defining features of what makes something archaea, is that it has ether linkage in its membrane lipids and not ester linkage. Another thing that can be different about archaeal membranes is, I guess I would say, what they'd look like overall. So here would be kind of, I guess from our standpoint, the most normal one. So it generally looks like what we think a membrane lipid looks like, except it's got an ether linkage. It's still got two fatty acid tails. It's still got a polar head group. What's different about this one that's really weird? Comparing to the top. Two blue boxes? Yeah, here's the phosphate head. Here's another phosphate head. All of this is covalently linked. It's a double-headed phospholipid. And that's weird. Or maybe we're weird. It's definitely different from what a lot of life forms have. So this has double heads. So does this have double heads? What's weird about this one? What jumps out at you out of that one?
The ring structures.
It's got ring structures within the fatty acid tail, which is unusual in the world of membrane lipids. So this one, double-headed, plus it's got ring structures. And what that means for the membrane construction is that here's, of course, of our normal situation and the situation that these would make, still a membrane bilayer. These two would not form bilayers because they don't need to. One lipid on its own has got two polar head groups. So they can go ahead and form a membrane, but this is all one piece, this is all one piece, this is all one piece. And the reason that this is helpful for these organisms is it makes their membrane more stable. and it's all one piece, it's harder for it to come apart. That's important at high temperatures. So all of those archaea, you saw their name is thermo this and pyro that. One of the ways that they hold up to really high temperatures is that they have crazy lipid monolayer in their membrane. So imagine heat thermally shaking this versus heat thermally shaking this. This is going to stay together better. But whether the membrane is bilayer or monolayer, whether it has ester or ether linkages, this is just a zoom in of what I just showed. The plasma membrane is still doing the major things that you've learned about before that plasma membranes do. So it's still a permeability barrier, keeping certain things out while letting other things cross. It's still a protein anchor, having particular proteins that help the function of the cell. Some of those are letting materials in or out in a selective way. And don't forget this one. I'm going to state this in a different way. Membranes are critical for establishing gradients. All kinds of gradients, but particularly gradients of ions. And those gradients of ions can then be used for chemiosmotic ATP production. So you've probably most recently learned about this when we talk about ATP production in the mitochondria. Remember that? You push a lot of protons to one side of a membrane and they flow back through ATP synthase and you get a lot of ATP production. Membranes of prokaryotes can also serve that purpose. They serve to separate ion gradients and then provide potential energy to then make ATP. It's just that in prokaryotes, it's their plasma membrane that is doing that job, not a mitochondrial membrane. And that will come up later in the class. Also, if you had cell last fall, this is probably still fresh in your mind. What can cross a membrane and what can't? If you haven't, you need to know that stuff that is small and generally nonpolar can cross a membrane with the addition of water, which is small and polar, but it still does cross membranes. Anything that's not small or nonpolar or water does not really cross membranes very well. So ions that have charges do not cross membranes on their own, nor do sugars, nor do amino acids, nor do proteins, because they're all either big or charged. So if a cell needs to get something in that is not small or nonpolar or water, it will have to have some special system to do that. This is where our break is going to be. If you'll stop it one more time, this will be a 3-video day.